Now, continuing the lecture, the cell wall. The cell wall is the outermost component seen in all the bacteria except the Mycoplasma chlamydia, that is not bounded by the cell wall but by a cell membrane. Spirochetes, Treponema, Borrelia, Leptospira species, they have got a flexible thin cell wall, a multi-layered structure lying external to the cytoplasmic membrane is the cell wall. It is composed of an inner layer of the peptidoglycan and an outer membrane. It varies in thickness and the chemical composition depending upon the bacterial type. Peptidoglycan of the cell wall give structural support and the forms, the shape of the cell, and the peptidoglycan cell wall is the only unique thing which is found in bacteria, not in any other species. So cell walls of the gram-positive and the gram-negative bacteria differ. Peptidoglycan in the gram-positive bacteria is thicker than the gram-negative bacteria. So the gram-positive bacteria retain gram stain, purple color of the gram stain, and gram-negative bacteria lose the gram stain. So some bacteria have structures external to the cell wall like capsule, flagella, and the pili, which are lying outside of the cell wall. Some gram-positive bacteria have fibers of the teichoic acid that protrude outside the cell wall. So here one can see the difference of the cell wall structure of the gram-negative bacteria and gram-positive bacteria. As you can appreciate, the cytoplasm of the both is present, then plasma membrane or the cytoplasmic membrane that is also present in both, then the periplasmic space, that is the space between inner cytoplasmic membrane and outer membrane, then there is thick peptidoglycan layer which is present in the gram-positive bacteria and also in the gram-negative bacteria, but the thickness is variable, more in gram-positive bacteria and less in gram-negative bacteria. Then the outer membrane that is containing lipopolysaccharide and the porin, that is only positive in the gram-negative bacteria, and here it is absent in gram-positive bacteria. Again, one can see the difference between the two cell wall. So the gram-positive bacteria, here one can see the fibers coming out from the cell wall. These are the lipoteichoic acid, and that is also the fibers coming out in case of the gram-positive bacteria, and here in case of the gram-negative bacteria, instead of the teichoic acid that contains lipopolysaccharide layer, that will be detailed in next few slides. But here one can see the thickness of the peptidoglycan layer, that is the thin peptidoglycan layer, and here is the thick peptidoglycan layer, and the cell membrane is of equal size present in that. Outer membrane, only the gram-positive, gram-negative bacteria, not the gram-positive bacteria, have this complex outer membrane. It's a major permeability barrier. In contrast, only gram-negative bacteria have a complex outer membrane, and it consists of the lipopolysaccharide, LPS, lipoprotein, and phospholipids. Its polysaccharides and lipoproteins behave as an antigen for the different gram-negative bacteria. Outer membrane takes part in the formation of the periplasmic space, as I have told you, that is the space between the cytoplasmic membrane and outer membrane in the gram-negative bacteria. This membrane contains porin protein which form a trimmer or the channel from where the nutrient can enter and leave the cell, which regulates passage of the small hydrophilic essential molecules like glucose and salt into the cell, and amino acids and vitamins and metals can enter through these porins. Also, many antimicrobial drugs such as the penicillin can also pass from this porin and can destroy the bacterial cell. Here one can see the outer membrane in case of the gram-positive bacteria that is absent, and here in case of the gram-negative bacteria it is present here outside this outer membrane, outside this cell wall, and here one can see the structure which contains the porin. It's a kind of channel, or it's a kind of canal or channel which allows the different things coming from inside to outside and outside to inside. Mainly it is from the outside to inside. Lipopolysaccharide, LPS, one of the major components of the outer membrane of the gram-negative bacteria. It is the part of gram-negative bacteria outer membrane. Only one gram-positive bacteria, Listeria monocytogenes, contain LPS. Otherwise, it is not present in gram-positive bacteria. LPS molecules have three regions: lipid A, phospholipid, responsible for the toxic effect of the gram-negative bacteria; core, the polysaccharides of the 5 sugars; and O antigen, it is the outer polysaccharide. LPS molecules are assembled at the plasma or the inner membrane, and the O antigen of this polysaccharide is exposed on the exterior, while the lipid A faces inside, and this is why the antigenic variation is seen in case of the gram-negative bacteria because of the O antigen. Now, what are the functions of the lipopolysaccharide or LPS? It produces endotoxin, kills humans, causes gram-negative septicemia, death due to LPS. It is just like a suicide bomber, and it is the part of the gram-negative bacteria which behaves like endotoxin, and that is just going to cause the death of a human being. Pyrogen causes fever, stimulates immunity, activates macrophages and the complement. It also induces interferon production in response to the attack. So that is responsible for producing interferon. Heat resistant and hard to remove. And appearance of the colonies is mucoid because of the presence of this lipopolysaccharide, and smooth depend upon the lot of the LPS or the capsule. So the colonies which look like smooth and mucoid, that is because of the more LPS and the capsule. It causes tissue necrosis and the tumor regression. Now, lipopolysaccharide, as one can see, the periplasm, cytoplasm, and here it is outside that is the gram-negative bacterial cell wall, that is the outer membrane which contains lipopolysaccharide, and here the detailed structure of the lipopolysaccharide. This is the lipid A, which is lying inside of the towards the inside of the cell, and here it is the O antigen that contains about 40 units, and that is antigenic properties of this lipopolysaccharide because of the, and the core polysaccharide which supports the lipid A and the O antigen. It is in the center. Periplasmic space, the space between inner and the outer membrane of the gram-negative bacteria, site where beta-lactamase and the other degrading enzymes are produced, or one can say the weapons of the bacteria to save themselves from the antibiotic and the other attacks of the chemicals. It is not seen in gram-positive bacteria. Here one can see the periplasmic space between the inner cytoplasmic membrane and the outer, and here one can see the periplasmic space between the inner membrane and the cell wall. So this is the space between, in case of the gram-positive bacteria, between cytoplasmic membrane and peptidoglycan, than between the peptidoglycan and outer membrane. Peptidoglycan, synonymous murin and the mucopeptide, these are found only in the bacterial cells, the unique property, as peptidoglycan is present in the bacteria, not in human cells. So that is a good target for the antibacterial drugs. So we do find the drugs which are capable of killing or dissolving the peptidoglycan. Ultimately, we are going to kill the bacteria by dissolving the peptidoglycan layer of the bacteria. Peptidoglycan gives shape to the cell and allow cell to withstand osmotic pressure such as the water. It is composed of the sugar backbone glycan, so the name, and the peptide side chain, so the name is peptidoglycan. The glycans is the main pillars, consist of the alternating N-acetyl muramic acid, or called NAM, and N-acetyl glucosamine NAG. So amino acids cross links NAG and NAM. D form of the amino acids used, not the L form, because the D form of the amino acids are hard to break down. So these are being used by the bacteria. Here one can see the main structure of the peptidoglycan. So that is the glycan made of the alternate NAM and NAG, and that is cross-linked by the peptide bridges. So for easy convenience, one can think like the ladder. So the two main pillars of the bamboos of the ladder, these are being connected by one piece coming out from this NAM and one piece from this NAM, and these are interconnected by the cross chain. So the muramic acid, D-amino acids, and D-amino pimelic acid are not synthesized by the mammals. These are only synthesized by the bacteria. Lysozymes kill bacteria by cleaving glycan backbone of the peptidoglycan. So the antibiotic do attack this peptidoglycan, and that is going to kill and cleaving the glycan and just breaking that stair and just making collapse the bacteria. Peptido side chains are cross-linked by the transpeptidase or penicillin binding protein PBP. This is the site being targeted by the antibiotic just to dissolve the peptidoglycan. It is an enzyme that is attacked, inhibited by the penicillin and the cephalosporin. So they do have this site of action. Peptide side chains contain D and L amino acids. The side chains are cross-linked by the peptide bridges. These peptide bridges vary in structure among different bacterial species. So one can see the glycan that is made of NAM and NAG, then that being connected by two chains, and these two chains are also again connected by one more chain. So the antibiotic do attack here, and this is also the place for the penicillin and the cephalosporin to attack here and to dissolve. So the glycan separates and the peptido is dissolved. So the ultimate result is the peptidoglycan have gone. Atypical cell wall of the acid-fast bacteria. Some bacteria do not contain peptidoglycan. Mycobacterium, like Mycobacterium tuberculosis, have unusual cell wall, and it is not trained by the gram staining. This is called acid-fast, as they resist decolorization with acetyl cold during the Zn staining. They have got the high concentration of the mycolic acid lipid in their lipid cell wall. This causes the slow nutrient exchange and the slow growth of these bacteria because of their high lipid wall. cell wall content. Then there are certain bacteria which has got no wall, so these are called wall-less form of the bacteria. These are formed when bacteria are treated with the lysozyme or the lytic enzymes, antibiotic interfering the biosynthesis of the peptidoglycan of these bacteria, or development T cell wall is not formed, so wall-less bacteria are produced. Usually these treatments generate non-viable organisms and having no walls. Four types of these non-viable wall-less bacteria are seen: Mycoplasma species, L-form, Spheroplast, and Protoplast. Wall-less form, the Mycoplasma, naturally lacking a rigid cell wall. These are the smallest living microorganism that can live independently. Then the second is L-form. They arise from the either gram-positive or the gram-negative bacteria, but they are unable to produce the peptidoglycan. So these are called L-form. Then protoplast, these are seen in gram-positive bacteria. Their cell wall is lost by the action of the lysozyme enzymes. And last is the spheroplast, seen in the gram-negative bacteria with a damaged cell wall, and these are called spheroplast. Damage is done by the toxic chemical or the antibiotic substance to just damage the wall, and the bacteria become wall-less. Here one can see the different form of the bacteria. There is the wall-less L form, and then there is protoplast, seen in gram-positive bacteria, lost the cell wall, and in gram-negative bacteria, when they have lost the cell wall, they become spheroplast. Teichoic acid, these are the fibers that protrude outside the peptidoglycan in some gram-positive bacteria. I have shown in the previous slide also that some fibers coming out from the cell wall. So these are the teichoic acid fibers seen mostly in the Staphylococcus and Streptococci, Lactobacilli, Clostridium, Cyanobacterium, mostly in the gram-positive bacteria. These are made of the glycerol phosphate or ribitol phosphate, so their names are different according to their contents which they do have. So classified as ribitol TA or glycerol TA. Some are linked to an acetyl moramic acid, NAM of the peptidoglycan layer. So these are also sometimes called wall teichoic acid. Some are linked to the lipid in the cytoplasmic membrane called lipoteichoic acid, abbreviated as LTA. Now, what is the function of the teichoic acid? They provide rigidity to the cell wall by attracting cations like magnesium and sodium, assist in the regulation of the cell growth by limiting the ability of the autolysins, do not allow the autolysins to destroy the bacteria, act as a receptor molecule for some gram-positive bacteriophage, and this help attachment of the staphylococci to the mucosal cells. By these teichoic acid act as adhesins, streptococcus pyogenes, induce septic shock like endotoxin, LPS in gram-negative bacteria. So these work like endotoxin LPS in gram-positive bacteria as the endotoxin LPS in the gram-negative bacteria. That is strongly antigenic and cytotoxic. Here one can see the teichoic acid fiber, as one can see these wall-associated protein. These are arising from the cell wall, and one can see the lipoteichoic acid that is taking origin from the lipid layer of the cytoplasmic membrane. For that reason, these are called lipoteichoic acid, and those which take origin from the wall, these are called wall-associated protein. Here again, one can see that lipoteichoic acid taking origin from the cytoplasmic membrane, and those which are taken from the wall, these are the wall teichoic acid. Here is a beautiful painting on the whole external wall of a house. It's a marvelous piece of painting, and really one do continue see it and continue to see it.